TopMedTalk.
All right, well, hello and welcome back to TopMedTalk here at Euro Anesthesia 2023 in Glasgow, Scotland. It is the annual meeting of the European Society of Anesthesiology and Intensive Care. It's TopMedTalk in the exhibit hall on the Edwards booth. I'm Desiree Chappell, and I'm joined by Professor Monty Mythen. Hello, Monty. Can I still call you that, Professor Emeritus?
You can call me whatever you want, within reason.
I'll be nice.
You can call me Michael if you want. There aren't many people who
Michael,
do that.
That is.
Yeah.
Your mum, maybe.
Yeah, she does.
Yeah, mom. And some cousins.
Michael. Desiree, it's great to be here. Good afternoon.
Good afternoon. Did I say good morning? I might have.
Whatever.
We've been having lots of good conversations here on TopMedTalk throughout the day in the exhibit hall. Monty, we've said it. We continue to say great vibe here in the hall today.
I think coffee speaks to people.
Oh, I think you're right. There's some really good coffee here. It's not like that everywhere.
There's a barista machine over there which has got the sort of glass walls that exposes the inside of the...
Oh, yeah, that's really interesting.
That's very cool, isn't it?
Yeah, it is very cool. So we'll put a picture up online so everyone can see that as well.
There's no actual coffee on the table in front of us.
Well, I'm sorry.
Where's that voice coming from?
So I was going to introduce all of our guests in just a little bit, but that demanding guest who wants their coffee. I'm just kidding. We'll get you some. Chop, chop.
It's my name, metabolic substrate. What can I say?
For all of us. Rupert Pearse, welcome back to TopMedTalk.
Thank you for inviting me.
Yeah, I'm sorry we don't have your coffee for you, but we'll get it before you leave. That's good. So Rupert, here at Euro Anesthesia 2023, once again, we were able to catch up last year in Milan. You're busy as ever this time around as well. I think you have like 4 or 5 different presentations.
3, I think.
Just 3?
You're scaring me.
Oh,
now.
maybe
I
you're
might
a
have missed a
panel.
couple.
I think you have I think you're supposed to be on stage right now.
And a lot of meetings, I'm guessing.
Yeah, catching up with a lot of people.
Yeah, lots of friends. Well, to introduce one of our new voices, Rupert, who wasn't here with us last year, Andy Cumpstey. Hello, Andy.
Hi, Desiree. Hi, Rupert.
Hi.
See you again.
Thanks for joining us. Well, Rupert, there's lots to discuss and lots to follow up from last year. First of all, Monty, where should we start, do you think?
I think we should dive straight into Optimize 2.
I think so too.
Potted history of where we got to the Optimize 2 point. The reason being, it's finished and we're expecting the results soon. We're definitely not getting the results today.
I was going to say, you can't share them, right?
No.
I don't know them myself, in all honesty, and I don't even have an inkling. So the statisticians that we work with, they work in a proper formal CTU, a trials unit, and they have like all sorts of SOPs they have to follow for crunching the data, so it has to be checked and checked again, and they get someone else to check it. So it's going through all of that process. So they probably know what the answers are, but
but we'll know when again?
Public will know on July the 6th.
The Thursday of EdPom, whatever date that is, July
6th. July the 6th.
But I think 2 weeks before we see them, then we've got to go through and sort of make sense of it and work out
I'm sure it's a
how
lot
we're
to
going to share the
unpack.
story.
Yeah. Wow.
Exciting
that's great.
times.
Congratulations. And how long have you been working on Optimize 2? And the team?
Crumbs, I can't remember. It's been so long because the trial spanned the pandemic, right?
Yes,
so we for I think at least 8 years.
Yeah.
On one way or another.
Yeah.
We've been trying to get the trial set up.
Yeah.
Then we're very kindly funded by Edwards, which really has made the trial possible.
Yeah.
And it's
with a grant.
With a major research grant.
Yeah.
So, and that's great. It's been great to work with a really positive relationship with an industry partner, with a common goal in terms of how we can get the evidence we need to decide exactly how we should use this technology. So that's been really helpful.
So go on then. I'll ask the obvious question for the listeners at home, Rupert. What is this technology and what's the trial looking at?
So the technology is a cardiac output monitor, in particular one that can give you a beat-by-beat measurement of stroke volume. And there's a bunch on the market, but the one that we use is the technology developed by Edwards, as that's they're funding the trial. We use their technology, seems fair enough. But it's transferable information. Anything that gives you a reliable, accurate measure of stroke volume. And we do 2 things with those data. The intervention will use the stroke volume measurement as an endpoint for giving fluid, as the most sort of robust way of knowing exactly how, if your patient's hypovolemic, do they need a fluid challenge? Yep. If they're not hypovolemic, they don't need fluid, and you can give less fluid. And that's part of the strength of what it's supposed to do. It's supposed to avoid overdose as well as avoid underdose. And then we supplement that with a low dose infusion of either dobutamine or dopexamine, sort of like a vasodilating inotrope, but quite a low dose, so 2.5 mics per kilo a minute of dobutamine. With the
it's a fixed dose.
Yeah. So
very pragmatic.
It is very pragmatic. Absolutely. I mean, the whole story is optimized too, as you know. I mean, I've been doing the same trial for like my entire career.
But you were in shorts the first time.
Yeah, exactly. Yeah, don't remind me. I mean, what I'd say is that we've refined it, we've perfected it, we worked out what was too much, we worked out what was not enough. We've shown that biological mechanism of effect. We know it improves tissue microvascular flow and tissue oxygenation. We know that if there's a benefit to it, it looks that's reducing post-operative infection. And that's where the benefit focuses. And we know that the people who most need that are older, high-risk surgical patients having abdominal surgery. So Optimize 2 has really pulled together, 15 years, nearly 20 years of research, and is sort of saying this is our strongest hand. This is the best intervention, the best type of technology, the right patient group, the right timing, and all of those things. So, in a couple of weeks, we'll know whether that does in fact have the beneficial effect on infections that I guess we all hope it will. But if it doesn't, we know that we've shut down that avenue of work, and we need to take a different approach to looking after these patients.
Yeah. And how many patients were you looking at?
So it's 2,502.
Oh.
Is the sample size.
Yeah.
And you hinted at it, but so these are all older patients having elective abdominal
major
procedures.
All over 65 having major gut surgery.
Yeah, as opposed to Floella, which we can probably talk about at a different time, which is similar intervention but in a different patient group. So this is elective surgery specifically.
Exactly. Elective major gut surgery.
Floella was emergency laparoscopy.
Exactly.
Yeah.
And the Optimize 2 website, because I've had a reason to look at it quite a lot in the last year or so, is an exemplar, I think, of how to have the information up online and accessible to see how it's designed, what the statistical plans are. I hope you agree.
No, well, well, thank you. I mean, that speaks to something. I mean, the way I see my role is as I'm a sort of the guardian of something on behalf of everybody, of the entire community, on top of patients, and we've got a responsibility as more older senior researchers to make sure that everybody gets to have a legitimate share in all of these things, and that means you've got to be really open and transparent. You've got to be inclusive in the way you build a trial team. So when Optimize 2 gets published, it won't be published with named authors. It'll just be the Optimize 2 study group, and so it's very much recognising that the people who worked really hard in individual hospitals to recruit patients, their contribution is every bit as important as mine and everybody else's. And I think that sort of equity in it is really important, and that's part of the whole thing of making that information there and available. And we have seen trials recently, some quite big trials, where people have kind of tweaked the protocol halfway through and kind of not really been very public about that. And I just don't think, if it was okay to change it, then it's okay to tell everybody. And if you can't hold your hands up and say, I made a mistake and this is what I've done, or, we did this for good reasons, then you've already got a problem because people can't interpret what you've done.
Yeah, and that definitely clouds the whole thing.
So a couple of the recurring things that we've discussed on TopMedTalk, and the trials community discusses it on a very regular basis, is the things that often leave trials dangling at the end of the day, is that the primary outcome event rate is typically lower than expected, or different to expected, but commonly lower. And the compliance with the intervention is not as anticipated or hoped. Were you allowed to have a sense of those two things as you travelled the journey? Because you can look at the whole event rate, can't you, and as opposed to by group, and say we look like we're on target.
Yeah. So, I mean, the problem with being a chief investigator is as soon as you start taking a little peek under the lid of what goes on, it's like Pandora's box. You suddenly know, and your ability to be objective and make objective decisions in the best interest of the trial becomes so much harder. And it's like, I don't know, when you're in a competition and when your kids are in the competition, do you judge them too hard or too soft? You just don't know anymore what fair looks like. So I'm actually really strict with myself that I don't get involved with that.
But can't you have people do that for you, if you see what I mean?
Yeah, for sure. I mean, sometimes you do need to do that.
Yeah.
You do need to look at the compliance and the event rate for valid reasons. We did a trial on post-operative CPAP called PRISM, where we did need to look at those things as we went along. But in Optimise 2, we've been able to not do that. One of the reasons we've been able to not do that is that we know from previous trials, we know from Optimise 1 that we have very high levels of compliance. We know we've got a deliverable algorithm.
That's one of the reasons I was looking at the website and looking back at the studies, because I was involved in some other trials where it was disappointingly low.
Yes.
And they were saying, it's always low. And I thought, well, it wasn't in Optimise 1.
No.
It was 90+%. I think it was 93%. Now, and in the paper, in the appendices, you put in some of the additional manpower that is required to achieve that level of compliance, but it's achievable.
Yeah.
Yeah.
So I don't know, but we do have our data monitoring and ethics committee, and they've been absolutely excellent all the way through. But at no point have they raised anything with us. Now, mostly they don't tell us very much, but you would expect as a chief investigator, if you were finding that you were running a trial that had such a low event rate that you were massively underpowered, it would be the DMEC's role to be coming and saying you may need to think about increasing your sample size or changing some choices. So we haven't had any feedback at all that makes me think that the event rate is any different to what we expected.
Did you have stopping rules both ways, or did you go for the let's plough through unless they say, look, your event rate's so low and the compliance is so poor, this is going nowhere?
Yeah, so stopping rules are interesting because they sort of swing way one way and another for various valid reasons in terms of how trial designs evolve. But the Optimise 2 has one stopping rule, and that is if we've got recruitment fatigue and we can't finish the trial, we're going to stop, and that's going to be the reason we stop. Because even if you do get a higher than expected event rate, all of other things, if you do stop early, it still does generate a form of bias that can actually mislead. So
I've got, before I hand back, I've got one other thing, and the other reason I was looking it up in detail again was the collection of a primary outcome, and the primary outcome is infection. The infection is still a composite, because it's a composite of infections.
Yes.
And the definition of those infections has to be rigorous and robust and reproducible and objective. So there's a lot written about that an impressive amount written about that in the protocol. And the second thing is it has to be meticulously collected by people who didn't necessarily see the intervention. And you also bought into the idea that you do a 30-day follow-up, which means you're committed to identify infection, non-infection for 30 days. Yeah. That's a lot of extra work, because sometimes what people do is they say infection and they report infections that were reported, which is very different to going to make sure that those infections aren't there.
Yeah, I mean, in any trial, there's a balance between getting the best possible, most robust data, and designing a trial that's easy enough to deliver that you can gain a very large sample size. And so you've got a sort of competition there between those two factors. And to some extent, if you get a really great sample size, you can cancel out certain problems with data capture. But ultimately, it's never as good as having a really robust, well-collected, well-defined data set. So we try to sort of trade off both a little bit. I mean, 2,500 patients is a reasonable sample size, something like this. There are bigger trials. There are a lot of much smaller ones. Yeah,
that's very true. Well, kind of switching gears just ever so slightly, this has been a big randomized controlled trial, something you've been working on for a very long time. We talked to Joyce Young yesterday about trial design, and that was something that we discussed last year. So let's talk a little bit about that and switch gears from RCTs to looking at other ways to answer questions or answer some of the questions that we have in our space.
Yeah, I mean, whatever we do, the randomized trial or the randomized whatever it is always going to be the ultimate evidence base, because what that does is it just takes away selection bias. And now lots of people are using epidemiology, these vast data sets we can now get out of healthcare systems. And you can get data on millions of patients with all sorts of fascinating stuff. But there's always what you would call unmeasured confounding. So there's always a risk factor that makes somebody have a worse outcome that just doesn't have a measurement that appears in the data. And there's some really interesting conversations you can have around, for example, the ASA score with that, because the ASA score is the most absurd score when you look at it. It starts out at one end asking how much comorbidity is the patient, and as it gets more severe, it's about what the anaesthetist thinks about whether they're going to survive or not, which is kind of switches halfway as to what it's asking you to measure. And I've got a colleague who's a nephrologist who I work with. He says ASA it's absolutely rubbish, but it's also the most powerful predictor of outcomes, because what it actually measures is not what ASA definitions are. It's just that anaesthetist's gut instinct about how this is going to go for them.
And nervousness.
And amazingly enough, that turns out to be a really powerful thing.
Yes.
So, obviously, we collect ASA score in Optimise 2, and we'll obviously adjust our results based on the balance of ASA score patients in each group. But if you get a big NHS routine data set, most of the time, there are exceptions to this, but most of the time, that won't have ASA score in it.
Oh, really?
Yeah, so you're not got that measure of the anaesthetist going, oh, crumbs, I don't look at this one, this is going to be difficult, we need to be careful here, we need an ICU bed. And so you have these patterns between groups, and patient, if you're interested in looking at epidemiology of a treatment, and you find that this group of patients seem to do better with this particular treatment, but there'll be a reason why they had that treatment that it doesn't, you don't get a measurement of. So you always want to end up having randomization, if you can. But randomized trials are really expensive, really time-consuming.
That's the whole question.
There's a lot of bureaucracy around research ethics, and every time you move an apostrophe on the protocol, you have to get an amendment. If it's a drug trial, you need a drug trial regulation as well. They're very, very tightly, very, very strictly regulated. But the hardest and most painful things about a randomized trial are, number one, setting it up.
Yeah.
Actually getting the protocol written, the database built, the statisticians happy with what you're doing, the data managers happy with what you're doing. All of that work should be doable in 6 months, but often takes more than a year. Yeah. And that really hurts you, because you're not collecting any data at all, and you're already behind on your deadlines, and your funder's already looking at you going, where's my trial?
Yeah.
The second big thing is recruiting and opening sites. And it is an absolute truism. I have never seen a trial where all of the sites opened on the same day as the lead site, that they wouldn't have finished on time.
Yeah.
And every trial I've ever been involved with, if all of the sites opened on day one, it would have been fine. But it takes ages to open those sites for various different reasons. So what we saw in the pandemic was an existing idea, something called a platform trial. Yeah. Well, you don't just have one trial answering one question, you shut it down, then you build another trial that looks almost identical, that answers a very similar question in the same patient group. And you just keep doing that over and over and over again, wasting, a year between trials as you go. And there was a very big trial, in particular trial called Recovery, which really completely defined COVID management in hospital. And there were lots of great trials in the pandemic, but Recovery is head and shoulders the most powerful, the most impactful, because it answered so many questions, and it had a platform design. So there was one trial, it had one protocol, it had one ethics approval, all the hospitals signed up to it, and periodically, we started with a question.
When you say all the hospitals, you mean all the hospitals in London, England, the United Kingdom?
Almost every NHS hospital in the UK took part in Recovery. And that's part of our centralised research funding system. The government were able to say... Total engagement, yeah. Now, there are other countries in part of Recovery now since the worst days of the pandemic. But so it's an international trial now, but those early data all came from the NHS. So early questions like hydroxychloroquine were answered by Recovery. It was the first trial to actually give us a definitive answer that hydroxychloroquine didn't help. Mm-hmm. It also gave us dexamethasone, which would have been a question, well, I didn't believe dexamethasone would work, and it proved me wrong very quickly. So there were lots of treatments. Tocilizumab was another success that those of us who look after septic patients were a bit sceptical about, but actually, when you get a big homogeneous patient group, it worked. But it ploughed through lots and lots of questions in a time frame that was remarkably short. And it did that because it just had one protocol, one set of ethics, all the sites were open, and then they added trial questions. And then bit by bit, one trial question would get answered, and they'd stop giving hydroxychloroquine to people, and they'd start giving dexamethasone to people, and then they'd stop giving dexamethasone, and then switch to tocilizumab. But they had several different trial questions going along in parallel. There were questions around aspirin and all sorts of things. Now, what that did was you didn't have to open and reopen sites. You didn't have to open and re-recruit them. You didn't have to get a new protocol every time.
Go through the hard yards
all those hard
of an
yards
RCT.
that really hurt trial delivery.
Yeah.
So we've all learnt from that, and now everybody wants a platform trial
Yeah.
for every question. And that's not necessarily right for every area of research, but it just happens that in perioperative care, you've got a very, very big population.
Yes.
Very, very big population. We in perioperative care, we still have not come to terms with that gearing.
Yeah.
We don't use that big population anywhere near enough, and that's very powerful. So very big
Just to
population.
put that in context, let's just talk about the size of the current backlog, because your group of in peri-COVID quantified some of the backlog for surgery.
Yeah,
That's not all the surgery, that's just the backlog.
Just the backlog.
How big's the backlog?
So in the UK we do
Millions.
roughly 5 million surgical procedures a year, and at one point the backlog was approaching 2.5 million patients.
Wow.
So, and
That
it
figure.
swings up and down for various
Yeah.
different reasons. So, but you get a vast population and a vast population of people waiting for the surgery they didn't get during the pandemic. And the other thing about perioperative care is there are lots of little things that we do, and they're not going to change the world, but they help. And they help a little bit.
Mm-hmm.
And so you're looking for very small effect sizes. You're looking for a very small advance in gain on patient outcome. And so you need very big patient samples for that. And so that means you need a lot of hospitals, and you need to not be opening and closing them all the time. And a lot of these treatments are not massively different from each other in terms of the nature. So what we need, and I think can really, really make more use of in perioperative care anesthesia than pretty much any other specialty in secondary and tertiary care, is just a really very, very large platform trial that's sort of owned on behalf of the whole community. That anybody can come along and say, I've got this question, can we do it? And it doesn't come down to the decision of someone like me or someone like Monty or someone like Andy. There's like a proper, objective, transparent process for deciding, is that a question that we want to answer? Is it a question that we can answer? Is there a funding there to support it? And so that nobody feels like that facility is owned and controlled, that they feel like it's on behalf of everybody.
So one, for example, very straightforward question, we'll just pick a couple of examples. TIVA versus vapor.
Mm-hmm.
That's a very common question, the mode of delivery of anesthesia. Here we've been discussing the last few days non-continuous versus continuous or near continuous blood pressure measurements.
Yeah.
A variation on current practices.
Mm-hmm.
All those sorts of things, you could drop them in effectively.
Very easily.
Yeah.
Very easily. I mean, it sounds too good to be true, and I'm always suspicious of things that sound too good to be true. But the truth is, the reason these trials are hard to do is because we've been making them hard.
Yes.
Yeah.
And we've been making them hard in an unnecessary way. There's a culture around running trials that if you compare practice in a clinical trial to practice in routine clinical practice, we cut corners every now and again. There are little things, right? We don't need to do that quite as, whereas in trial you have to, because you're somehow...
Yes.
So the same sort of lapse in a clinical trial is treated very, very harshly compared to routine practice. That doesn't make any sense. And so all of that sort of culture means that we make it much harder on ourselves to deliver these trials.
Yeah.
And the platform trial just takes that away.
And I think we're going to be hearing a lot more about that in the years to come.
Yeah.
And I think we're going to be hearing a lot more from you, Rupert, and your friends back in the UK, which is great.
Always happy to chat.
Before, I know the others want to ask you about your other trials that are ongoing at the moment, or a couple of them anyway. And we've talked about this a few times before, and I've been harping on about it recently. When you did Optimize 1, you took what they call a frequentist approach to the powering and the analysis.
Yeah.
And you were just shy
Just to
of the...
interrupt that, Monty, can you explain that
did
for
you wanna
listeners at home? What does
Let's
that
call it
mean?
the traditional...
We're going to
the
divert
traditional
here for a
approach,
minute, Rupert, so you can...
where you make an estimate of what you think the primary outcome event rate is going to be and the effect size.
Yeah, exactly.
You do a power calculation,
Yeah.
which is often, we talked about this yesterday, often compromised by the fact that you want to do 2,000, but you've only got the money for 800.
Yeah.
So can you squeeze the outcome variable a bit, and can you tweak that to make the numbers work? Then you get to the end of it, and people say, well, there was no difference. And you look at it and say, well, actually, there was a really big difference.
Yeah.
But it didn't have the magic p-value of less than 0.05, the frequentist approach. Now, I think Rupert was involved in a subsequent publication because I think the ASA, not the American Society of Anesthesiologists, but some statistical group, contacted you and said if you'd used a Bayesian analysis, it would have been a highly significant result. What's that all about? In other words, if you'd chosen a different way of looking at the data, we'd all have said, oh, well, that one's sorted.
Yeah. I mean,
And that's all published, isn't it?
You
It's
could
all
have a very long
published.
conversation about this.
That's all published, isn't it? It's all out
It
there.
is. It's all in
May
the
require
public domain,
beers.
and it's a fascinating conversation. But the basic thing is, why do we do science? Well, I mean, there's discovery science where you invent new things, but if you put that to one side, all the rest of science, it's about eliminating bias.
Yeah.
All the scientific methods we do are trying to go, we want to find out what's really happening, but we can't study every single molecule or every single organism in the world, so we have to take a sample. And when you take a sample, you're at risk of bias, and good science reduces that risk of bias as low as it can go. And part of all of that is, how do you decide, have I found something or have I not found something? And you set yourself rules. You set yourself some rules before you do the trial that says, well, if I see it and it looks like this, I'm then going to call that the drug works.
Yeah.
And if I don't see it passing that threshold, I'm going to say it doesn't. Now, there's a big debate around, what as you quite correctly call Monty frequentist design. That's when we have like a p-value, and it's p less than 0.05 means it works, and p above 0.05 means it doesn't. But of course, all the p-value is, and I'm not going to get too bogged down in how you define a p-value because that's a whole controversy of its own. but really, all the p-value is just a guide to how likely you are to be wrong. And you can be wrong both ways. So just because it appears that your trial is not significant doesn't mean that you weren't measuring a real biological phenomenon.
Yeah.
It's just your rules that you made for yourself at the start have said, well, your rule that you made before you did the trial says this doesn't work and this is what it looks like, so your drug doesn't. So we then go around calling these things negative trials. And I don't think there's any such thing as a well-designed negative trial on an important question. That's absurd. But everybody wants a success story, don't they?
But that's simplistic. If it was a 100-meter race, your intervention group, if they're running the 100-meter race, you cross the line first, if you see what I mean, but you only crossed the line 9 meters in front, whereas we said it didn't count unless you crossed the line 12 meters in front.
It's
Is
kind
that
of
about
like
it?
that. It's kind of like
and then
that.
people say, well, it was a draw.
Yeah.
So hang on a second, I was 9 meters in front.
I mean, that's not an unfair analogy. But now, if you can take a different approach to that, and you can say, well, look, we're not comparing a brand new drug that we've never used before in human beings that could have all sorts of serious side effects. We're comparing two ways of saying, how do I decide how much fluid I give? Because you're going to give fluid to everybody, right? So how do I decide how much? And, vasoactive drugs, well, we give them to everybody as well, sometimes infusions, sometimes boluses, but we give them to everybody. So how do we decide that? So we're not talking about giving a completely different treatment in one group to another. So what you really want to know is, on balance of probability, which one works? And so what Bayesian statistics do is they tell you that this one is probably better than this one. And here's our measure of probability. And in the analysis you're describing on the optimized data, the probability was, I think, 88% probability that the goal-directed therapy intervention is better.
I think it was more like 98%. But anyway, you look it up again. But
I have to go and reread my own
I gave
paper.
a lecture on it recently and thinking, well, whatever way you look at it, it looks like they said it's really highly likely that difference is real.
For sure.
But in between, even though it said it's really, really likely that it was real, we carry on doing mainly the other thing.
Yeah. So there's a caveat to all of this. And that is that you've always got to worry about harm.
Yes.
in any trial. And one of the things that we historically have done very, very badly is measuring harm and understanding that treatment harm is a different thing to treatment benefit.
Yeah.
And the problem with using mortality as an outcome measure,
It's
the ultimate composite outcome
Yes.
measure, is it reflects both harm and benefit. And so we've forgotten, actually, that we need to measure specific harms of the treatments that we use. And then when you come to a Bayesian approach and you're looking at mortality, and that's that composite of benefit and harm, we sort of kid ourselves that there is no harm because it probably reduces mortality. You've got to ask yourself, are we confident it's not doing harm? And so there's, in my view, we need to be really, really careful which trial questions we use Bayesian stats for. And in all of the trials we do, we've got to have really robust data on harm.
So before I hand off, in the interim, I've been involved in a number of discussions, including with regulators, where they seem to accept the idea that you would state a priori, as we say up front, that even if you're using a frequentist approach, you reserve the right to do a secondary Bayesian analysis to determine. It doesn't turn the p-value into a new magic number, but it says it's okay to say it didn't achieve that p-value, but look. So are you going to do that? Or with optimized
we haven't predefined the Bayesian analysis. I mean, our DMEC chair is Tony Gordon, who's the king of Bayesian stats, right? So
he's probably run the numbers already.
He probably already knows, yeah. I mean, I think what we got. Why are we having this conversation? What is this all about? And ultimately, it's like we're asking a philosophical question, and we're trying to predefine what the answers look like, so that we don't, when we get the answer we didn't want, we don't rig it.
Yes.
Yeah. That's why we predefine all of these things, so that we don't go, oh, actually, I wanted a different answer. Do we think if we nudged it a bit, would that be okay? It's like, well, no, because if you're nudging it, that's kind of sort of like lying, and we kind of sort of can't do that. So we've got to predefine it, and then we create this framework, this rigid framework for ourselves, that because we want something robust, and it's all about that philosophy of your trial question. And you've got to match the philosophy of your trial question to the statistical methods that you're using. And if you don't do that, you end up in a pickle. So,
Andy, you're a researcher. I'm sure this is all.
It's fascinating to hear it from your. I mean, you've done so many trials over the years and so into the detail of all this and got it on your fingertips. But I wonder what the listeners at home understand about Bayesian analysis.
Well, they're not going to learn it from me.
I was going to say,
how is that going to interpret the results, though? When you say, I get what Monty's saying, we're running this 100-meter race, but how can someone reading that at the other end have confidence that, okay, I analysed it this way, I got one answer?
So to interrupt for 1 second, I highlighted it recently. The New England Journal of Medicine does have a very nice little 6-minute cartoon that they published recently, which is trying to understand rapidly the difference between the frequentist and the Bayesian approach. And it uses an analogy, I think, of whether it's quicker to take the train or to bicycle to work. Yeah. And it still leaves one with a lot of questions, but I thought it was a pretty darn good attempt at explaining the difference between the two. Sorry, Rupert.
I mean, but basically, you've got one that just gives you a hard yes or no.
Yeah.
And you've got another that gives you like a continuous scale, like a thermometer. That 0 degrees is almost certainly not, and 100 degrees is almost certainly. And it could be anywhere in that. And we can have a conversation about how probably we would like to see. But the problem with the frequentist, that old approach, is it's just yes or no. It's either positive or it's negative. And that's a very destructive way. Because most trials, the results are very nuanced. They need a sophisticated interpretation.
So coming back to the question about RCTs and adaptive trials then, and you mentioned about, you said about we cause our own problems with this culture of RCTs. Who's we?
Well, there's a whole industry of
We's
people.
we, brother.
There's a whole industry of folk.
Interesting question.
So you've got, I mean, one of my jobs is I run clinical research for my hospital trust and for my university. So I see everybody's trials running. I also support our research governance team. They're the people who check that the ethics and the MHRA and all of those things are ready before it goes off to the committees. And I also do my own trials, so I'm a sort of poacher and gamekeeper. And I've also got a role with one of the UK funders of research. So I see the research coming through the funders.
Yeah.
And if you look at it from all these different areas, there's a sort of culture that spans all of those domains, that exists in the clinical trials units, that exists in the ethics committees, also in the research governance officers, also to some extent in the funders.
Yeah.
That is slowly becoming stricter and stricter over time. And that often frustrates the entrepreneurial chief investigator because I can't believe it. Why don't they just let me do it? I don't understand what the problem is. And sometimes my main message is there's two sides to every story. Always, when you hear those arguments, it's always the chief investigator hasn't done something very well, and the governance person's being too strict. There's always that kind of balance. There's always a bit of both that needs to change. But anyway, when I have a conversation with our governance folk, it's always like, okay, there are two things I care about in my job being head of research. Number one, what's the patient risk?
Yeah.
So you're having an argument with your chief investigator. What's the patient risk?
Yeah.
And if I get a blank look of, well, there isn't one really, okay, all right, fine. What's the organisational risk?
Yeah.
Is this thing that we're doing going to be something that gets us on the front page of the Daily Mail for running a corrupt organisation or whatever it might be? And if the answer to both of those questions is no, then really, why are we even talking about this? Why can't we just be pragmatic about it and get on and do the research? If the answer to one of those questions is yes, that's quite serious.
I was thinking about.
But a lot of investigators do get very upset with the governance folk when actually they just don't do their job very well. And a lot of governance folk do get very upset with investigators when they're just being too strict.
Yeah.
So,
great answer.
Rupert, you've been so generous with your time. One more question before we leave. Last year, we were talking about some other projects, which I don't know how you do all of these, do all at the same time. Some other projects that you're working on. You want to give us an update on some of that?
Yeah, I mean, I didn't because I work in a big team of amazing people who I'm very proud of. So I don't really do any of the work. I spend my time sitting in conference booths having lovely conversations with my friends. But no, so there's a few things going on. I mean, something that I've been really passionate about, which we've managed to keep going through the pandemic, is a program called ESAIC. And that's looking at shared decision making for older high-risk patients having major surgery and trying to understand better ways of helping them make a decision that they are happy with, that they don't regret later on.
Yeah,
and that's what's been really interesting about that is rather than just doing a clinical trial, we've been doing qualitative research and interviews. We've been doing epidemiology. We've been doing sort of what we call discrete choice experiments, where we get people to pretend that they're a doctor or pretend that they're a patient, and sort of answer questions about what they would do in a particular scenario. And the whole output of that is that we know that the high-risk surgical patient now, as a group, they tend to think very differently to younger people having much more straightforward surgery, where the harms of the surgery are often very related to the disease that they've got.
Yes.
Whereas high-risk surgical patients, the harms of the surgery are rarely that the anaesthetist made a mistake or the surgeon made a mistake.
Yeah.
It's much more common that they just got pneumonia or they just had a myocardial infarction, and it wasn't really related to anything anybody did, and it would be very hard to predict. And so those off-target complications are difficult to explain to people. And often they can't understand why, well, I've got arthritis of my hip. Why are you talking to me about my heart?
Yeah.
Yes.
So our hope is that we've learned through that understanding, through that early research, we've designed now what we're calling a forecast model. And it doesn't try and predict the future, because I don't think that's really possible. What it does is it says, these are the things that you need to think about. So, it's like a weather forecast. It's really, really sunny out. I know that if I don't put suntan lotion on, I'm going to burn. But other people don't need to put suntan lotion on because they've got lovely tan skin. Yeah. And so we make an individual decision based on those data that we get. And hopefully, I won't get sunburn, and nor will my friend, and neither of us will regret the decision. But, I mean, we'll see. We'll see. I mean, it's very possible that having that extra information doesn't make any difference to patients at all. And if that's the case, then we can probably change our guidance for doctors and be much less strict with doctors who are very, very worried that they're not giving patients enough information. So that's a big one. That's called ESAIC. We're also doing a lot of work in Africa with a colleague, Bruce Bickard,
Yes.
in Cape Town. And there's a really amazing team of folk in Tanzania, Uganda, Ethiopia, South Africa, who are doing research around failure to rescue after surgery.
Oh, yeah.
After surgery in African hospitals, and also maternal hemorrhage after cesarean
Yes.
section. Also trying to build a big routine database, starting with Uganda and South Africa, which will be really exciting. The idea of having routine data in that context is something that, till recently, nobody thought was possible.
Yeah.
And probably the first thing that you'll see is we're doing a big epidemiological study of critical care provision across Africa. And that one's called ASIOS. It's literally just about to start.
Yeah, it's amazing.
So we're very excited about all of that. So those are some of the things we've been up to.
And penguin, you mentioned earlier on, was it?
Oh,
Yeah.
yes, penguin, of course. So penguin
I
is...
used to love a penguin. Chocolate biscuit, was it?
Indeed,
Double
indeed.
chocolate
You
there.
don't see them anymore, do you?
You don't know. I don't know where that came from.
It's very
So-
British, apparently.
Yeah.
Well,
We don't
anyway.
have those.
Yeah, okay. Well, we can go on and have a conversation about who has the best confectionery. But and Penguin is a very, very large randomized trial of 80% oxygen versus 30%
Yes.
oxygen to prevent surgical site infection. And
In which patients and context? Sorry,
so this is patients in lower middle income countries from about 12 years up who are having open abdominal surgery. So whether it's elective, whether it's emergency, but open abdominal surgery. And we've got sites in India, we've got sites in Nigeria, we've got sites in South Africa, we've got sites in Mexico. We're opening sites, I hope soon, in Brazil, Uganda, and Tanzania as well. So it's a very big trial. We're about 4,000 patients through with a 13,000 patient target. So modest in size. We've got a little way to go. We've been really struggling to get people to fund it, actually. It's been really
Interesting.
difficult. We've had fantastic support from a team of surgeons in Birmingham who run a group called Global Surge. It's not just based in Birmingham, but the key people who've been helping us with it are in Birmingham. And they've been amazing in really bankrolling it. But it's been very hard to get external grants. So if anybody's got £500,000 or even 100,000.
One to give out.
We could do a lot of good with that money for Penguin because it's been very hard to get it funded. But the hope is that that the chlorhexidine mouthwash maybe will prevent pneumonia. But the oxygen question, I think, is a really interesting one, and Andy is the absolute expert on this. But there's a very controversial set of guidelines from the World Health Organization saying that we should be giving all of these patients a high concentration oxygen after surgery to prevent surgical site infection. And those guidelines primarily, not exclusively, but primarily driven by infectious diseases doctors. Yeah. But here's the thing, oxygen infrastructure is the most difficult thing in every hospital.
Yeah, absolutely.
And in low-income countries, oxygen infrastructure is unbelievably expensive, very, very difficult to maintain. And you really, really do not want to divert resources in a very resource-strapped healthcare system and spend it all on oxygen because of a World Health Organization guideline that a lot of people think is based on very tenuous evidence.
Yeah, absolutely.
So, we'll see what we see. I mean, it's an objective trial. The answer will be what the answer is. But I think we need now to really put a marker down that we shouldn't be putting guidelines like that. Because in low-income countries, the World Health Organization guidance is very powerful for that.
Yeah, absolutely. Rupert, thank you so much.
Thank you.
We really do appreciate it.
It's been lovely chatting.
It has been lovely chatting. We can't wait to hear in a couple weeks, a month, at the EBPOM meeting. Will you be there, or the
So I'm actually at our trainees meeting in Leeds.
Yeah.
So my colleague Mark Edwards, who's Deputy Chief Investigator in Optimise, is going to get all the fun and present the results. And I'm going to join online and just give the tale of woe, which is my 20-year journey in goal-directed therapy research.
Enjoy that.
Or AKA Pearse's folly. But,
and it's going to be live broadcast.
It's going to be live
live
streamed,
podcast,
which is great. And thanks a lot for the EBPOM team for helping that, because it's really important our investigators here at first.
It's really important for you to be there too. So I'm glad we could do it. EBPOM July 5th, did we say the Thursday? 6th. I'm sorry, 6th. July 6th, the Thursday of EBPOM. We'll be live streaming that.
4th,
5th, and 6th. That's right.
American Independence Day.
Yeah.
The beginning of the National Health Service,
Yep.
and Optimise 2.
There you go.
4th, 5th, 6th.
I wonder where you were going with that.
All right, Rupert. Again, thank you so much.
It's
And
a
thank
pleasure.
you so much for listening to Top Med Talk. You can find all these great conversations at topmedtalk.com, on social media, Twitter, Facebook, LinkedIn. We are there, and we're on your favorite podcatcher as well. So catch up with us. Thanks so much, team.
Top Med Talk.
Thanks for downloading Top Med Talk. Don't forget to subscribe via your podcatcher. Don't forget to check us out on social media. We're on Twitter, Facebook, LinkedIn, and YouTube. And also, don't forget, Top Med Talk is the broadcasting arm of EBPOM, Evidence-Based Perioperative Medicine. We'd love you to find out more about that. If you check out ebpom.org, you can find low prices on some of the conferences we're organising around the world. Many of them are virtual and don't even involve you leaving your own home. Check out ebpom.org now.